In this video we will discuss about the matrix stiffness in cancer and the normal ECM. So basically we will discuss the overview. Detailed explanation we have discussed in the last few papers that we have covered. So in the normal ECM we can see that everything is separated apart. However, when the matrix stiffness increases, what it will does, it will leads to the increase in the collagen, and this increase in collagen will further leads to the matrix stiffness increase. So this will cause a formation of vicious cycle, and thereby along with that what it will does is the matrix stiffness increase, it will also leads to the increase in the fibronectin, and this fibronectin increase will affects this RhoA pathways. So this RhoA pathway is the important mechanosensor that helps in the interactions between this matrix stiffness and the tumor cells. The RhoA pathways, I think it's not shown here. Along with that, there is also increase in this TNC expression. TNC expression leads to the formation of one protein, that is tennessine. It is also not shown here, and this increase in tennessine will leads to the mechanically activated tumor promoting environment. And before this tennessine, it is just a stiff matrix, but the tennessine is expressed, then it will lead to the activated tumor promoting stiff environment that will helps in the tumor proliferations. And then this is about the fibroblast. So fibroblast will also be activated in the stiff matrices, and because of this RhoA pathways, what it will does is it will leads to the epigenetic changes within the tumors in a way that it will promote the survival pathways such as NF-kB pathways. And along with that, this about the macrophages. What it will does, the macrophages when it goes into this tumor microenvironment or in the stiff matrices, it will differentiate themselves towards M2 type macrophages, which is tumor promoting in nature. So basically it will use the immune system of the host for its tumor growth and survival. Along with that, this T cells, what does the T cells is the suppression of the T cells will occur because of this checkpoint inhibitors receptors that are expressed on the T cells. So this checkpoint inhibitor receptors will be neutralized by their antigen that are overexpressed in the tumor microenvironment. Thus the immune suppressions will also take place, and simultaneously as the matrix stiffness increases because of the contractions, it will also provide a physical barrier that will create additional hurdle for any drug to reach to the tumors. Thus it helps in the chemo resistance. Apart from that, the tumors also generate a environment within the cytoplasm that it will excrete or it will remove most of these chemotherapeutic drugs when they reach the cytoplasm, that we have discussed in our previous video, that when we are discussing last year of the previous paper. And other things that is mentioned here, there is cross-linking that we have discussed as the increase in the cancer-associated fibroblast occurs because of the increase in the matrix stiffness. It will also promote the contractions, and that will also cause this collagen cross-linking. So and this all variations occurs when the stiffness of the matrices increases, and this results in the changes in the signaling pathways, not only in the stromal cells but also in the tumor cells and the immune cells that are in the tumor microenvironment, that infiltrate the tumor microenvironment. And so this is how the matrix stiffness plays a role in the cancer, and how critical is matrix stiffness for the cancer or the tumor proliferations and survival. So it could be one of the therapeutic targets to reduce the matrix stiffness so that tumor cells become sensitive towards the chemotherapy, other cancer therapeutic approach such as chemotherapy or CAR T therapies or other or the combination of dual therapies. That's all for this video. If you need to understand more about this matrix stiffness in cancer and the normal ECM differences, you can go through the reference that has been provided in the description. Thank you for your time.